No.
Okay. Oh. Okay. Welcome to the Grant Rounds. I'm very proud to introduce Dr. Corinde Wiers. Please let us know in the chat or the Q&A if you have any questions. We'll be monitoring that, or if there's any technical troubles. And I have one housekeeping item, which is that the CME code is 5656, which should also be posted in the chat. So it's my great pleasure to introduce Dr. Wiers. She started her career in the Netherlands at the University of Amsterdam, getting a master in psychobiology, and then continued in Germany at Freie Universität Berlin to get a PhD on addiction research. And then went on to work with Dr. Nora Volkov at the NIH and studying neuroinflammation, glucose metabolism, dopaminergic and glutaminergic function. So doing a lot of imaging using PET, for example. Some of her top cited papers from her PhD are on cognitive bias modification and how this can be applied in smoking cessation or alcohol use disorder. And some of her top papers from her postdoc are on dopaminergic function and how it relates to socioeconomic status and gender differences. And so her work has always impressed me because it's very applied. She always thinks about what this means for real life situations and how to integrate this into treatment. And so she recently started her own lab at University of Pennsylvania. Her current research is primarily funded on developing treatments for alcohol use disorder using the tools that we have to study brain function. And I'll let her explain it in more detail herself. Thank you.
Thank you so much, Anna, and thank you all for inviting me here today in snowy Minneapolis. And it was very interesting when you just said what I researched before in my postdoc, that the words bias, gender, socioeconomic status, in a way I'm relieved I'm not studying that as per today, because those three terms would already be the blacklist in the Trump administration of being funded. Oh, this is going to be on YouTube, isn't it? Oh, well, anyway. Right now, yeah, working on alcohol use disorder at the University of Amsterdam, University of Pennsylvania. So just short background, alcohol use disorder, worldwide, 3 million deaths every year still occur as a result from harmful alcohol use. And over 1 in 7 Americans are estimated to develop an alcohol use disorder in their life. And despite these very large numbers, fewer than 8% of adults who need treatment for alcohol use disorder receive treatment. And that's really varies the reasons why. It's just access to treatment, but also there's still a lot of stigma in the field of addiction for needing treatment for a substance use disorder. So there's a need for interventions that can promote long-term abstinence and reduce alcohol craving that are easily accessible. So yeah, when I worked in the lab of Nora Volkov, which was, I always felt like a kid in the candy store at the NIH in Bethesda, Maryland. There were so many research opportunities, but we studied a lot of brain glucose metabolism using PET in people with alcohol use disorder, and also did acute challenges of alcohol. So what we found was that when people drink alcohol acutely, so it doesn't even have to be a very large dose, but we gave people alcohol, that brain glucose metabolism acutely goes down. And this was already a phenomenon known for many years, and it was first interpreted as, oh, you see, when you drink alcohol, the brain doesn't function well. There's less brain energy in the brain. But the measure of this strong reduction of about 20% in glucose metabolism never really correlated well with impaired cognitive functioning. So then in 2013, Nora did this study on the effects of acute alcohol on brain glucose and and on brain acetate. And she found like, oh, it's not just a reduction in glucose, but there's really this metabolic shift that when you drink alcohol, the liver breaks down alcohol and converts it into or metabolizes into acetate, and acetate can cross the blood-brain the blood-brain barrier and can be used by the brain or will be used by the brain as an alternative to glucose as an energy substrate. So when you drink acutely, the brain just shifts from glucose to acetate as an energy source. And she also found that people who were heavy drinkers, so were used to drinking alcohol, they had this metabolic shift stronger than people who hardly drank. So the brain is also to some degree used to this metabolic shift when you're a heavy drinker. And also during sobriety, people who are classified as heavy drinkers, so I think these are people who drink more than 20 drinks per week, the standardized drink, they show higher levels of acetate in the brain compared to social drinkers. Plus, very, very well characterized that during sobriety, heavy drinkers show lower brain glucose metabolism. So they can eat sugar or carbs as much as they want, the brain will not utilize it. And that persists for about 2 to 3 weeks after abstinence when the brain somehow normalizes again. So this hypoglucose metabolism is to some degree similar to, for example, Alzheimer's disease, where you also see strong reductions in glucose utilization of the brain. But in alcohol use disorder, it looks like it's more temporary. But still, there might be a relevance to alcohol use disorder treatments, namely that during prolonged detox, so when people come in, if they have a very heavy alcohol use disorder and need alcohol detoxification or withdrawal management to treat their alcohol withdrawal symptoms, it might be that this shift in brain energetics, because the alcohol is not there anymore, so the acetate is not there, and the glucose metabolism is quite low. So there might be this energy deficient state in the brain in people with alcohol use disorder that may contribute to withdrawal symptoms that we see, such as anxiety and tremors, and could even lead to seizures, as well as alcohol cravings and relapse. It might be, oh, my brain needs energy. I crave alcohol to make my brain feel normal again from an energetic perspective. And a ketogenic diet has been hip in the last 10 years, I would say, just as a means of reducing weight. And also for more like reducing cognitive fog, that people feel like much sharper when they're in a ketogenic diet. But it's basically a diet that is very high in fat and low in carbohydrates. And it has been over 100 years now that it has been used for the reductions of seizures in people with epilepsy. And there's still clinics in the United States for people who don't respond to first-line treatments for epilepsy. That's longer-term outpatient ketogenic diet interventions actually help. And the diet leads to this nutritional state of ketosis, namely the cells in the body are all using ketone bodies for fuel, and the liver produces the ketone bodies rather than glucose. And these ketone bodies are acetoacetate, beta-hydroxybutyrate, and acetone. And here below you see the diet composition of a ketogenic diet. Like the classical one is really a 4-to-1 ratio of grams of fat to grams of carbohydrate and protein combined. So it's really like 80% fat, 20% carbs and protein. So it's almost like drinking oil. But it has been shown now also that if you do more like a 1-to-1 ratio or a modified Atkins diet, that also leads to ketosis and is much more sustainable for longer-term treatment, for example, people with epilepsy. So yeah, the goal of my lab right now is to see whether ketone bodies could help overcome this hypothesized energy deficient state in alcohol use disorder. So rather than getting this brain energy from acetate through alcohol, can we give ketones, particularly acetoacetate, is very similar to acetate structurally, can we give ketones almost as a replacement therapy in people who have low glucose metabolism? And in 2018, there was a first study by Anders Fink Jensen's team in Denmark that did an alcohol withdrawal study in alcohol-dependent rats. So they basically put alcohol dependent, or they made the rats alcohol dependent, not so difficult to do, they love alcohol, and quit the alcohol exposure and treated the rats either with a ketogenic diet or just a standard high carb chow diet, and found that those rats on a ketogenic diet demonstrated lower body rigidity and irritability. I'm still not quite sure how that translates to alcohol withdrawal symptoms in humans, but still this was the first study that showed, yes, there might be beneficial effects of this ketogenic diet in or during alcohol withdrawal in an animal model. And when I was still at the NIH in Bethesda, Maryland, as part of my K99 R00 grant mechanism, so I was still a postdoc, we performed a first 3-week ketogenic diet trial in alcohol use disorder inpatients. So patients came into the hospital. There was a small alcohol detox unit where we were able to treat people. But the NIH, it's a research hospital, so people were also part of research studies. So we basically treated a little bit over half. I think we had like a 2-to-1 ratio. So more people were randomized for the ketogenic diet than to a standard American diet. And the standard American diet is just, I think, I don't remember, it's a high carb diet, I think more than 50% carb. And it was very important to me that for this trial that it would be blinded, because if we would give people oil to eat, his very strict ketogenic diet, and other people spaghetti and tomato sauce, it would be too easy to know what diet you were on. So we worked with the metabolic kitchen at NIH. This was really phenomenal, and they developed dietary shakes. So people are randomized to receive a dietary shake, breakfast, lunch, and dinner for 3 weeks that was either ketogenic, high in fat, or standard American, high in carbohydrates for 3 weeks. After our first patient, we realized we can't just do a liquid diet. People really missed chewing things, so we gave ketogenic snacks on the side. Like what do we give, like cucumbers, etc., and like chicken salads or so. But people were relatively okay with this diet. But it's also, these people were very, very severe, people with alcohol use disorder. So already before they came into the hospital, most of their calories came from drinking alcohol. So this liquid diet probably is the easiest for people with alcohol use disorder compared to others. But these figures show, just as a proof of principle, that our ketogenic diet group, 19 individuals, they increased ketone levels in the urine. It was tracked every morning by an independent researcher. And the standard American diet, which apparently is more commonly abbreviated as SAD. I should be doing that instead of SA, the SAD diet. They did not have any ketones in their urine. And the same with blood. We just, on the day of MRIs, we did an MRI every week. Ketone levels were rising. There's a bit of a dip in the 3rd week. I think there were 3 patients who went on a pizza party and couldn't resist eating pizza other than their dietary shakes, which is understandable. We were also able to detect ketones in the brain. We used proton magnetic resonance spectroscopy at 3 Tesla and used a very standard sequence. So it was a little bit of a shot in the dark. We saw elevated levels of acetone and to some degree acetoacetate, but we're not able to detect beta-hydroxybutyrate. We're working now at 7 Tesla and also more edited sequences to to detect ketones much better than this in the brain. But still, you were able to distinguish whether people are on a ketogenic diet or the SAD diet. But the most important, I think, finding of this study was a clinical outcome, namely patients were prescribed GABAergic benzodiazepine medication to reduce their withdrawal symptoms. And they always were prescribed the benzos when withdrawal symptoms were high. And we saw an interaction effect of the diet times the time in the first week when withdrawal symptoms are high, such that the group of the ketogenic diet required fewer benzodiazepine medications to help their withdrawal management compared to the standard American. So basically, the diet may have contributed to reductions in withdrawal, therefore required fewer benzodiazepine medications. And also the number of people who required benzos was statistically lower in the ketogenic diets after the diet initiation compared to the standard American diets. But of course, this is an extremely small sample size, 19 versus 14. This has to be replicated. Nobody has done this study yet. I mean, it's quite an intensive study to do this for multiple weeks, but still, it will be needed. So yeah, as I said, weekly we did an MRI scan. We also did an alcohol cue-induced brain reactivity test where with functional MRI, we projected pictures of alcohol and of appetitive food to people and of neutral cues, such as a very neutral lamp over there or a neutral bridge. And the idea was to look at more dopaminergic regions or reward-related areas. Does the ketogenic diet also dampen alcohol cue-induced craving in the brain? And we actually analyzed it. There was a really interesting paper by Hedy Koper that came out 2 years ago in Nature Neuroscience that used machine learning based on several cue reactivity studies previously acquired, where she basically developed a neurobiological craving signature, namely these brain areas in the ventromedial prefrontal cortex, ventral striatum, and medial cingulate cortex. They were elevated in people with substance use disorder looking at substance cues, and they were associated to explicit levels of, yes, I'm craving this when I see this. So what we did for this data set, well, first we looked at behavioral ratings of wanting. So people saw all the cues outside of the scanner and said, okay, how much do you want this picture? And there was a small effect of that the ketogenic diet led only in week 3 to a reduction of wanting alcohol cues compared to the control or the standard American diet. But when we looked at this neurobiological craving signature, there was a main effect, namely the ketogenic diet had a lower neurobiological craving for alcohol signature compared to the control diet. But it was throughout the 3 weeks. And it might be our first MRI was done when people were already in ketosis, just when people come into the hospital. We had to clear them, do inclusion-exclusion with them. We had to look for their liver panel test, etc. So it took some time first to randomize them. We randomized them to the diet within 2 days, but then also for them to feel well enough to undergo an MRI scan. I think the average day was on day 5 or 6 when ketone levels were already high. So it's not too surprising that it wasn't an interaction effect, but a main effect still needs to be replicated, I would say. And in parallel, we worked with Leandro Vendruscolo, who runs his lab at NIDA in Baltimore. He's a preclinical researcher. We wanted to understand, well, doesn't ketogenic diet also affect alcohol consumption? So he works with Wistar rats. They love alcohol, and really first trained them on a chow diet to self-administer alcohol. Then rats were randomized, or no, half of them were provided a ketogenic diet and half a chow diet for 2 months. And initially, he wanted to look here for alcohol self-administration, but he changed the design a little bit, and I'll come back to that after. That actually in this experiment, and also just again, proof of principle, ketone levels were high in the ketogenic diet group, and glucose levels were actually lower. But he put the rats back on chow for 8 weeks when they were, then half of them were being made alcohol dependent with these alcohol vaping rooms, and half of them were vaping air, so they were not being made alcohol dependent. So now we suddenly had 4 groups: history of ketogenic diet, alcohol dependent; history of ketogenic diet, control; history of chow diet, or no keto, alcohol dependent or control. And what we found was many different lines, but basically all the way to the right, you see that those rats that had a history of a ketogenic diet, they basically had lower levels of self-administration of alcohol compared to those with a history of chow. So this was really the first study that showed like, oh, wow, even a history of a ketogenic diet could reduce alcohol consumption or self-administration in an animal model. And that has been to some degree replicated. In 2022, there was a lab in Spain that showed that mice who were on a ketogenic diet acutely also self-administered alcohol less. And that's also what Leandro did recently. We're still working on this paper, but yeah, again, rats on a ketogenic diet, I think here it was 8 weeks, self-administered alcohol less compared to those on a chow diet. And there can be many mechanisms of this effect of a ketogenic diet in reducing alcohol withdrawal, alcohol self-administration or consumption. And there's this really good review of Christopher Palmer, who is a psychiatrist who works at Harvard, and he treats people with schizophrenia with a ketogenic diet and has success with that. So our background really comes from this PET background and glucose metabolism and this whole idea of a shift in brain energetics. So really more like, okay, a pathway would be this glucose hypometabolism, which you also see in Alzheimer's. So a ketogenic diet can be very, very useful in people with Alzheimer's when glucose doesn't work, but their ketone metabolism is intact. But he actually summarizes all the other mechanisms as to which a ketogenic diet could be fruitful in all these psychiatric and neurological disorders, including mitochondrial dysfunction, definitely disturbed in alcohol use disorder, oxidative stress, definitely disturbed in alcohol use disorder, inflammation, definitely disturbed in alcohol use disorder, and glutamatergic GABA imbalances, definitely disturbed in alcohol use disorder. So this kind of opened a path, like, wow, there's so much to be studied, not just in all the other psychiatric conditions, but also in alcohol use disorder. Oh, and I helped Chris a little bit to say that it's not alcohol use disorder is also a psychiatric condition. So I need to tell him that in his next figure, he should add it also as a condition in his figure. But at least he cited us. He didn't completely take us out. But there was one other potential mechanism that we basically found completely incidental in rats, namely in the original experiment from Leandro, he wanted to see, okay, rats, 6 weeks, ketogenic diet, we're giving the rats 14 hours of alcohol vaping, and he was shocked the day after to see that all the chow rats were kind of happily like, yay, under the influence of alcohol, whereas the ketogenic diet, most of the rats were passed out and some of them actually died. So he saw that blood levels of alcohol were much higher, 5 times higher in rats who were treated on a ketogenic diet. So somehow rats cannot break down alcohol. The effects of alcohol are much stronger for them. And these are rats who were, vaping alcohol for 14 hours. That's not something that humans do. So we repeated the experiment just with IP, intraperitoneal injection of alcohol. It's also not something that people do, so we actually still need an oral experiment. But at least also it was not just the vaping. Also, if you inject alcohol, there is an effect, not as drastic as 5 times higher, but a small effect of higher levels of blood alcohol. So this was after 2 weeks of KD. So we really still need to look at all the differences: vaping, oral, IP, and the duration of alcohol exposure to see when does this happen. And also, why does this happen? Why can't rats break down alcohol when they're on this high-fat diet? Oh yeah, and this was one more level. Like, so the blood levels were 5 times higher, but also if Leandro set up this clever experiment that's based on blood alcohol levels. He gave less alcohol to rats to create the same blood alcohol levels. So basically, rats on a ketogenic diet required 40% less alcohol vapor to reach the same blood alcohol levels. And one answer that might be like, well, how? Why is this happening? It comes from the livers. So he dissected the livers of rats exposed to a ketone diet for 6 weeks and found that protein levels of alcohol dehydrogenase 1 that are extremely important for the breakdown of alcohols. If you have high enzymatic levels, you can break down alcohol into acetaldehyde and then into acetate. They're much lower in the ketone diet. So if you don't have that enzyme, your blood alcohol levels will be higher because you can't break it down. And we're still writing this up in a paper. I think I've been presenting this for over a year now, but one day it will be published. But there may be other mechanisms too, we don't know. So in the interim summary on the ketogenic diets, we demonstrated that a ketogenic diet intervention study in humans with alcohol use disorder during alcohol detoxification, that this 4-to-1 very rigid diet was pretty well tolerated. The diet reduced benzodiazepine intake and a neurobiological alcohol craving signature, and the diet decreased alcohol consumption in rats and mice models. It increased sensitivity to alcohol, or it is blood's its pharmacokinetic effect, which might be due to alcohol dehydrogenase. And currently my postdoctoral fellow, Cindy, she is working on following up on that pharmacokinetic effect because it hasn't been demonstrated in humans. If you go online and look on Reddit, for example, where people describe their experiences if they are on a ketogenic diet for various reasons, for weight loss, they describe it. It's I can't tolerate alcohol anymore. Like 1 glass of wine or beer already knocks me out. But nobody has done the experiment. So Cindy set up a study. We have now only 7 healthy volunteers, or just healthy volunteers who drink. So they're more like healthy students where they undergo a 4-day ketogenic diet and a 4-day standard American diet in randomized order. And after each diet, we give them a fixed alcohol challenge based on their sex and weight to go up to a breath alcohol level of about 0.08, the legal drinking limit, which it's so fun to do. These experiments in the lab, everybody is just cheerful in the clinical settings. And the hypothesis here, or hypothesis, would be that the ketogenic diets would increase breath alcohol levels. And yeah, so the ketogenic diet led to elevations in ketone levels. I mean, I believe that the minus 240 is just when people come in after that 4-day diet exposure. And then this is just throughout the day, ketone levels decrease after alcohol exposure. We don't know why, but again, this is only 7. We're not done with the experiment. But we don't see the drastic effects with the 6-week diet intervention in rats when they were vaping 14 hours. We don't see this with an oral exposure in humans, but it might just be that our 4-day ketone diet is just not long enough, and also the diet that we give is not a 4-to-1. It's much more this modified Atkins of like 50% 50% of fats. So we'll finish this, but we wanted to go up to N of 15. But we need to develop a study that's just a longer intervention. And we also piloted the effects of the ketogenic diet in this group on brain glutamate levels, again with spectroscopy. We use now 7 Tesla, and together with Ravi Nanga, we are working on an edited GABA sequence. And in 7 people, there was no effect, but it looks like it's trending up a little bit as hypothesized. But yeah, we'll see once the study is over to see if there's a bit of a clear. And we also haven't really correlated it with ketone levels yet. It might be that some people who are not the most truthful on their diet. And again, longer exposure may also be interesting to look at. But then there are many limitations to a ketogenic diet. I've tried it many times and I quit many times. It's so hard to stick to. It's very rigid. If you eat a little bit of sugar, you're immediately out of ketosis. Or if you eat a piece of bread, you're out of ketosis. And if you drink a beer, you're out of ketosis. So it's hard to maintain, and it has low retention rates. So even I visited the neurology clinic at Hopkins, where Dr. Mackenzie Cervenka, she treats people with epilepsy for years with this modified Atkins diet. And even in people who know that they can have a seizure, for them it's also still very difficult. And I spoke with a patient who was seizure-free for, say, 5 years and wanted to just have a piece of chocolate, immediately had a seizure again. So even for people who are at risk of seizures, it's hard to stick to a diet. So is this really going to work with people for alcohol use disorder? We don't quite know, but it's going to be hard. So I made a little bit of a switch from ketogenic diet research to research on an exogenous ketone monoester. Also, I mean, it's been called a ketogenic diet in a bottle, but it's basically a drink that was developed by Dr. Richard Veitch and Kieran Clark, who work at both the NIH and in Oxford. When was it? 2012 was the first development of this drink. So basically, the exogenous ketone monoester, you drink it, and BHB or ketone levels are within half an hour are elevated without the need for a liver to produce ketones. So you're basically just drinking the ketones. And here you can see some of the kinetic plots. So this is delta G, that's a ketone monoester. So in half an hour, you reach BHB or ketone levels up to 3 millimolar, which is very similar to 2 to 3 weeks of this very rigid diet. So it's so much easier to elevate ketone levels by drinking this energetic drink compared to this rigid diet. And the first application of this ketone supplement was done in 2015. Richard Veitch treated a patient with Alzheimer's disease with a ketone supplement. And this is actually before this ketone supplement was on the market commercially. So he got in huge trouble for treating people with his ketone ester out of his own lab. He passed away a few years ago, so I can say it. But NIH was definitely, how dare you give this to humans when it's not approved yet by the FDA, or it has like an FDA GRAS position. But now it's commercially available. So there are various kinds. So ketone ester is a delta G, but more commonly now are the 1,3-butanediol drinks, like Kinetic, and we have Juvenescence that taste a little bit better. Delta G really tastes like motor oil. And then there are ketone salts. They're not as effective as elevating ketones as much, but they all basically lead to rapid elevations of beta-hydroxybutyrate or ketone levels in blood. And then, 2019, Brianna Stubbs found that, this ketone supplement also decreases appetite or desire to eat. So the current projects that are related to alcohol use disorder that we perform at Penn are a whole branch of effects of ketone supplementation on, can we also see this rapid shift in brain energetics using PET and MRI. Can we also see the effects of alcohol on alcohol pharmacokinetics? So you get more sensitive to alcohol when you drink this ketone supplementation. Does it help for alcohol withdrawal? Can you, rather than sticking to this rigid diet during alcohol withdrawal, can we just give people a drink for during breakfast, lunch, and dinner in addition to their normal carbohydrate high food? And also we're now hoping to look more on the effects of the ketone supplementation on alcohol consumption and brain metabolism. I have a pending NIH R01 and submitted the just in time, but yeah, with the current administration, it's unclear if grants are awarded. So we'll see. Just not in time, maybe. But yeah, the first study I did when I moved to Penn a few years ago was this PET study, where we invited 10 participants, 5 with an alcohol use disorder, 5 controls, to come in twice, once baseline after a night of fasting, and once when they drank, or after a night of fasting and they drank this ketone supplement. And we chose one that didn't contain sugar because for a glucose metabolism scan, it will just throw the balance of. It's better that everybody is fasting, and then we increase the ketones to have the glucose level stable between both conditions. So this delta G is just black because it didn't contain even a little bit of sugar compared to the blue one. So the hypotheses here were like similar to that alcohol map that I showed earlier. If you drink alcohol acutely, brain glucose metabolism goes down, acetate goes up, major shift in brain energetics. So we expected to see that too with this ketone supplement. We didn't have access to PET with ketone metabolism at that time. We do have now, but basically a reduction in glucose metabolism was expected. And we also would expect that this is stronger in our alcohol use disorder group of 5 compared to the healthy control group of 5. Namely, could it be that the individuals with alcohol use disorder, they're used to alcohol, they're used to acetate, might they also be more familiar, they have a greater affinity to alternative energy sources that are similar to acetate, such as these ketones. And also that it would go vary with reductions in craving. So yeah, so when people are fasting, that doesn't look good. Well, but when people are fasting and drink this drink, then blood BHB levels really skyrocketed. So it was like 4 millimole. We gave a pretty high dose, and people are really gagging when they drink it. They did not like it. And for those individuals with alcohol use disorder, we saw a statistical drop in alcohol craving, but it might also just be that they got nauseous from the drink and therefore didn't want to drink alcohol. We definitely need to look at that. And it's 5 people. And very similar, this is a brain glucose metabolism map. At baseline, people have steady levels of glucose, and it really dropped by 17% with this ketone supplement administration, like an hour before the PET scan. And the strongest effects are in very similar areas that are affected by alcohol. So if it's, as you can see, it's just all over the brain. But we did not see any differences between groups. So the reductions in glucose metabolism were similar in the alcohol users and the healthy control. So we can see like, okay, there might not be a greater affinity for ketones, but we can also see it like, okay, their ketone metabolism is intact. Glucose metabolism, maybe not, but ketone metabolism is intact. And we did some measures with spectroscopy. This is at 7 Tesla, where it was very easy with an edited sequence to detect BHB after drinking it. But it's not as fancy. Like, ideally, I would give people ketones or ketogenic diet, do a PET with glucose metabolism, and then a PET with carbon-11 acetoacetate, that you can measure both glucose and acetoacetate in the whole brain. You can really correlate one with the other. So if glucose goes down in one region, ketones go up, is there like a net increase in a specific region that might be therapeutically beneficial? But still, we were able to detect it. That's basically what we could do with spectroscopy. And then the study that we did on the effects of ketone supplements on alcohol pharmacokinetics. So it's very similar to the study I described earlier. Cindy led this study as well. So 10 healthy volunteers came in twice. They basically drank this ketone supplement, and then half an hour later, they received a low to moderate dose of alcohol. And on another day, they received a placebo and a low to moderate alcohol dose. And there we hypothesized, similar to a ketogenic diet, that the supplement would increase alcohol sensitivity, so increase breath and blood alcohol levels, and that it would lead, therefore, to lower wanting and liking of alcohol. But to our surprise, we found the exact opposite. So the KS plotted in red, the ketone supplement, we saw dampened breath alcohol levels, dampened blood alcohol levels with the ketone supplement. So completely opposite to ketogenic diet, but still we saw reductions in alcohol liking and wanting. So yeah, we weren't sure if this was, did something happen to the breathalyzer? Do the ketones do something there? So we asked Leandro to do the same experiment in rats. And of course, he just did it within a week, even though for us it took a year to acquire this data. And he also saw that Kinetic, which is the ketone supplement that we use, dampens the blood alcohol curve in rats. We were a little bit worried whether the sweetener allulose, whether that caused this effect, but allulose alone did not. So it's really the compound of the ketones that seem to drive this. But this was oral. So we were wondering, like, is this an effect on the liver? Does it somehow promote alcohol breakdown by promoting alcohol dehydrogenase? And we're really excited, like, if that's the case, we could give this to people who just overdose, and maybe it will help them accelerate their alcohol metabolism. But Leandro basically did a comparison between oral ketone exposure in rats compared to an injection. Oh, sorry, no, the ketones were always oral. The alcohol was first oral, so you have to absorb the alcohol first, versus the IP alcohol, where it's just basically injected in your bloodstream immediately, and then there's no effect. So there must be an effect on absorption. And now our working hypothesis is basically that these ketones lead to a delay in the absorption of alcohol, likely to a delay in gastric emptying, and which might actually be similar to what we see in these popular Ozempic GLP-1 agonists. Also there's a delay in gastric emptying. People don't like to drink on it. And it might be that when they drink, alcohol doesn't get absorbed. So if you don't get the immediate spike of alcohol in your bloodstream, it's just not so fun to drink, or you don't really like the effect. But yeah, there's a lot of research that still needs to be done there. And then I think last slide for the rats is that if we both provide oral and intraperitoneal ketones to rats, it immediately reduces their alcohol self-administration. But also there, it might just be that they didn't like the effect. So then they don't feel good, and therefore they don't drink alcohol. So we need a better control condition for that. And then the last study route is can the ketone supplement also affect alcohol withdrawal? And again, Anders Fink Jensen studied this in Denmark, and he compared the ketogenic diet with the ketone ester and showed that this ketone supplement was sufficient in reducing alcohol withdrawal. So that was promising. And we also found a very old paper from 1976 from Science that already showed that when rats in the dotted line were undergoing alcohol withdrawal, so at time point 0, they had no alcohol in their system anymore, that those who were exposed to 1,3-butanediol, so that's one compound of a ketone supplement that converts to BHB, it rapidly decreases withdrawal scores in these rats. And nobody did anything with this because this compound wasn't on the market for humans. And it took us some time to find it because we were never looking for 1,3-butanediol. We were always looking for ketones and beta-hydroxybutyrate. But 1,3-butadiene is just a compound that's in these ketone supplements that converts to PHP. But yeah, no human study was done. So we set up a study in collaboration with the Care and Treatment Center, which is about an hour and a half outside of Pennsylvania, and randomized 48 patients to receive this ketone supplement for breakfast, lunch, and dinner. Because if you drink the ketone supplement, it's only up for about 4 to 5 hours. So to receive a steady state of ketones, you would have to drink it 3 times a day, or a placebo for 5 days. So we really wanted to target that acute withdrawal phase. Like, does this have this much of an effect, or at least a similar effect as a ketogenic diet? And we actually just received the data and don't really see much. So if you look at the benzodiazepines that patients received, no group difference. If you look at the CWA, which is the clinical alcohol withdrawal scale, so it just basically rates the alcohol withdrawal symptoms in people, no major difference between placebo and ketone supplement within those first 4 days. And it might be, we're now actually looking that we may have a bit of a flaw in the design, because in alcohol treatment units in or inpatient care in the United States, there seems to be like a different approach to treatment. So at NIH, patients would come in and were only treated if their withdrawal symptoms were higher than a score of 8 on this CWA questionnaire. So then they would only be treated if withdrawal symptoms are high. So we could use the need for benzodiazepine as an outcome measure. But here, everybody who came in were put on a benzodiazepine taper based on the withdrawal symptoms at admission. So when everybody already received a fixed taper at admission, then you can treat them with whatever you want. It's fixed, so you can't really do anything. So you kind of really use it as an outcome measure. But still, I think we should have seen a difference in CWA scores if there really was a true effect. And yeah, it's also, if you compare the benzodiazepine use that we did in the ketogenic diet trial, there's much more variation between patients, namely the standard errors are much higher, and also the levels were lower. So basically, people just were massively treated with benzodiazepines in this treatment center. Therefore, withdrawal symptoms are high, so we can medicate them with anything or give any intervention, we might not see it. That's, yeah, possible. But we also did not see an effect on alcohol craving between placebo and ketone supplements. And we're still going to look more for like % difference if we take into account that baseline level of taper, but it doesn't really look that there was any effect. So an interim summary on the ketone supplements, we saw that it rapidly shifted glucose to ketones and as a brain energy source. We saw that the supplement reduced alcohol absorption and decreased alcohol wanting and liking in healthy volunteers. And it reduced alcohol craving in that small N of 5 non-treatment seeking patients with alcohol use disorder. And to me, it's really interesting that if you reduce alcohol absorption, it decreases wanting and liking. But then with the ketogenic diet, if you have this massive increase in blood alcohol levels, it also reduces alcohol self-administration. So it might be if you target the alcohol pharmacokinetics on both sides, rats, humans may not like it, or there are just all the other mechanisms that come into play with brain energy, etc. And in our more recent study, we did not see effect of the ketone supplements on alcohol withdrawal or alcohol craving in AUD inpatients. But we might want to study that again in a CWA base, or where in an alcohol inpatient unit where the benzodiazepines are being described based on symptoms, not as a fixed taper. So in conclusion and future directions, ketones might be of therapeutic benefit to individuals with alcohol use disorder by improving brain energetics or other mechanisms, including altering alcohol pharmacokinetics. We really have no idea yet about an optimal dose of the supplement or an optimal diet composition. What is the minimum level of ketones that are needed to reduce consumption or reduce alcohol withdrawal? And also, particularly for the supplements, a palatable product. So the first generation of ketone ester tastes like motor oil. Like we use it for the PET study, and I never want to use it again because people are really gagging. Horrible taste. The kinetic was a lot better, more like a sour lemonade. And a more newly developed ketone supplement tastes kind of a little peachy, milky, not too bad, but none of them really taste good, that's for sure. And more studies, I have a whole series of research questions, particularly long-term interventions of both the ketogenic diet and the ketone supplements on alcohol consumption. What happens if we give this to people who want to reduce their drinking for, say, 6 to 12 weeks? Does it reduce their consumption? Does it reduce their craving? Does it improve their cognition? Because suddenly they have brain energy source that is not alcohol or not having the detrimental effects of alcohol that might improve their cognition. And also we're working on other brain energetic outcomes, including more mitochondrial health, like NAD plus, and PET scanning with acetoacetate. So the PET scanning with acetoacetate would look a little bit like this. There's a Canadian group that is led by Stephen Cunnane, who has done a lot of research in Alzheimer's disease. So he showed, this is actually in a healthy control population, but he showed that if healthy controls are on a ketogenic diet for several weeks, glucose metabolism goes down, but acetoacetate, so ketone metabolism, goes up so much more that the net level of brain energy would increase, and particularly in people with low levels of glucose at baseline, such as Alzheimer's population, aging population, and alcohol use disorder. So this would be a dream study for me to do a dual tracer PET study after long-term interventions. And then we, of course, have this whole boom of GLP-1 receptor agonists particularly in this country, and or the Ozempic, or there are various versions now, Mounjaro, for example. And it was Anders Fink-Jensen who did all the preclinical studies on ketones, but he already warned me a few years ago, who cares about ketones? Nobody wants to stick to that, and particularly not in substance use disorder populations. And he was one of the first who showed how effective GLP-1 receptor agonists were in preclinical, but also in clinical populations with substance use disorder. People who are on this lose a lot of weight, but they also lose, not everyone, but many of them lose the drive to drink. And probably when they drink, alcohol doesn't get absorbed. So it's not fun to drink more or interesting to drink more. So it's also a research field that will probably be booming in the next few years, not only for obesity, but also for substance use disorder, including alcohol. And there's a whole, I don't know, some people say it's like, oh, the panacea, also for Alzheimer's, it seems to work, improve cognitive function, etc. But yeah, many thanks for your attention, and many thanks to the members of my lab, particularly Cindy, who did all the human experiments, and my collaborators, Leandro Vendescolo and Nora, still advises me, thank goodness. And yeah, thank you. Let me know if you have questions.
Okay, so there's currently no questions in the chat, but please feel free to post them. And I don't know if there's questions in the room. And I guess I'm just gonna pass this.
Thank you so much for that amazing talk. What a thrilling story of your research. It's just really fantastic. I'm Katie Perelman. So my question was kind of back to the beginning about the acetate, the finding that Nora Volkow found about acetate, and why does the brain, so I was thinking, why does alcohol cause this shift? Why does alcohol do that? And I think the answer is because it makes acetate. And in the presence of acetate, the brain prefers to use acetate instead of glucose. So my question is, why does the brain prefer to use acetate instead of glucose? And similarly, if acetate's available, why does it prefer that? And similarly, why would it prefer ketones over glucose?
That's actually a very good question. Why does it prefer? We just show that it does. But what we didn't do, so in these PET experiments, particularly the one on FDG, people are fasting. So it's a little bit unfair to do fasting versus a high caloric load of alcohol or a high caloric load of ketones. So it might just be like the ketones and the acetate are being pushed in the brain, whereas apparently glucose is being more pulled into the brain, but that pulling doesn't happen if you're fasting. So what we've never done is actually giving isocaloric glucose and isocaloric ketones, and look what it does to glucose metabolism. But you can't really do that with the brain metabolic rate of glucose. So it might actually still be interesting. Yeah, I don't know how we could do that.
Okay. And then,
but we still see that the utilization of glucose, goes down. So maybe that pushing into the brain basically gives that effect.
and then maybe relatedly, because you were saying how maybe taking ketones, these like supplements, somehow improves the metabolism of the energetics, but maybe it's, I don't know if improving it is similar, or is it's making it more similar to how it's offering something to that is like readily available, or it's making it like an alternative to whatever it might prefer, like instead of acetate. Maybe it's making it something available so that it won't choose acetate because it can choose ketone instead of glucose if it must desist from using glucose. That might be a very weird
can you repeat that again? So maybe we should develop something that rather than using acetate, the brain should use ketones. Is that
maybe? Yeah, I guess. Yeah, I guess I was just trying to understand why having ketones why would that be like improving the energetics of the brain?
Yeah, so in the case of alcohol use disorder, it's because there's this longer lasting low levels of glucose utilization. So even you can give people lots of glucose, but the brain doesn't use it. But if you give them ketones at that time, the brain does use it. So then suddenly your brain functions because there's energy that you can use. So I think the question is also, why doesn't the brain utilize glucose after long-term alcohol exposure? And actually, I don't know. I don't know why that's the case. Like, yeah, because it shifted to acetate. But why can't it rapidly change back? I don't know. It just is. I'm sure there are many researchers, preclinical researchers, who are working on that.
Anybody else?
Kasia Litak here. I'm addictions medicine and psychiatry. I'm not a researcher, so just observational comment is like when I worked in the substance use treatment center, most people withdrawing from substances, the most popular destination for them after lunch or dinner or breakfast was the vending machine with Snickers, bars, and all that. So everybody was basically loading on glucose. So it's interesting that you think that the brain doesn't metabolize glucose, whatever, the vending machine was toppled every so often because people just really wanted to get into it. So that would be something also interesting to explore, and the effects of the ketones on dopamine, which are believed to drive the substance use disorders, and whether there are applications for other use disorders rather than alcohol.
Yeah, I think it's a very, very good point. And it's very common to see this in alcohol use disorder, also opioid use disorder, also characterized by low glucose or lower FDG or glucose metabolism. It might just be an effect of this energy deprived state that you're just trying to fuel your brain with lots of glucose, but it's not effective and you're just eating more and more of it. But it may also be a mu opioid receptor component to all of it, to craving sugar during acute withdrawal. But it's definitely, yeah, we saw that also. It was really hard in inpatient settings to keep people on that ketogenic diet when there was Snicker bars, cake, etc., available all the time, and people were cheating all the time. So, oh, I thought this was a sweetener, but no, that's a sugar bag. We're not using that in this study. And the effects of ketones and dopamine. I think there's one preclinical study that shows that elevations in ketones lead to a dopamine spike, but nothing in humans, and also much lower than anything like. The sugar alone would increase dopamine more than ketones do.
But that would be for implications for treatment per se. That would be an interesting thought about giving people ketogenic diet in addition to SIWA. And one of our colleagues in the department is developing helmets for cravings. So, people drinking ketogenic diet and wearing his helmets. It's just an image that might be interesting for people to entertain.
Yeah, and I think in general, to optimize diets in treatment centers, particularly in the United States, would be amazing. I worked before, it was more in cognitive bias modification training in a treatment center in Germany. And there the clinic director was very keen on very healthy diets for everyone. So I saw people just after they were admitted and 3 weeks later after withdrawal, and people looked so much healthier, also just with much improved nutrition. So, yeah.
Okay. So we actually do have a question from the chat now. Jasmine Kamshong asked, is there any specific parts of the brain where glucose energy source changes with ketogenic diets or supplements?
So it changes almost everywhere, but I think the strongest changes were in the prefrontal cortex. So that's also something we actually haven't really looked at, like regional effects. Where is it most important? Yeah. So not sure, because we only had 10 people, and it was such a strong effect of 17% reductions.
Okay.
But at least for the field of alcohol use disorder, so there's this hypometabolism, and particularly the hypometabolism in the prefrontal cortex is mostly related to dopamine D2, low dopamine D2 level, or receptor level availability. So there is a correlation also there with the dopaminergic system.
Does glucose metabolism, does it return to normal functioning with longer abstinence?
Yes, so it's about 2 to 3 weeks that it returns to normal, thankfully. So it's not like in Alzheimer's where it's almost permanently damaged. But then there are studies that show that, correlations at least, or that's maybe increased exposure to alcohol might lead to Alzheimer's disease, but I'm not quite sure if the glucose metabolism is related to that.
I was curious because in relation to your point with people in recovery just wanting a lot of sugar, and then I wonder if that kind of persists into almost a food addiction. I mean, I have a family member who he recovered from alcohol use disorder many, many years ago, and I remember he was just constantly eating ice cream cookie sandwiches instead, but the craving for sugar has persisted. So I was wondering if that's just more of like a personality thing, or have we kind of switched from one addiction to maybe a, I guess, less severe addiction towards sugar and food in the process?
Yeah, so I think particularly in the acute withdrawal phase, it may be related to this hypometabolism, like, oh, the brain doesn't have energy, sugar, please give me energy, that doesn't reach, it's eating more. But it also might be associated with dopamine. So one characteristic, and it's a particular NORA studied over the last few years, dopaminergic system, that low levels of dopamine D2 receptors in the striatum very much predict substance use. So it's kind of also chicken and egg. If you have low D2 receptors, you're more likely to develop substance use disorders and obesity, also overeating. But also if you take dopaminergic agents, cocaine, but also alcohol that indirectly increases dopamine, that also leads to a downregulation. So the more you take the drugs, the more you want it in that sense. And that might be similar with sugar. But the findings in obesity are not so consistent as in substance use disorder. So that longer term effect might more be a dopaminergic effect.
Hi, I'm Kate Powell, a psychiatrist. Are there any negative health effects of the ketogenic diet and any difference between that, the ketogenic diet and the ketone supplement?
So we actually didn't measure cholesterol, but I think so cholesterol spikes in the first few months on a ketogenic diet. So that is being seen as an not so great or not effects of a ketogenic diet. But if you look at longer years, it actually normalizes again, even if the ketogenic diet is continued. The 4:1 ratio, this really high level of fat, is not recommended for most people. Maybe only epilepsy, severe cases, like children with epilepsy can be with a tube through the nose, can be fed like that to reduce seizures. But in general, it's not really recommended for a full balanced diet. I think an Atkins diet, just lower carb, would be beneficial for almost everyone in the Western world, but not the full ketogenic diet. So yeah.
I have a follow-up question to that. So is this like a threshold thing where, you know, you have to have a certain ratio in order to have some effect? Or is it a dose response, like the more fat, the better? Like, so you know, if I did like if I change my diet from 80% carbs to 50% carbs, am I going to see some effect?
So for the ketone researchers, they would say, no, you have to always be at a steady level of ketosis. So I've heard like this 1:1 ratio might be the minimum. But already there was a study done in the Canadian group in an older residency home with people with dementia, and they were all put on a low carb diet, not a ketogenic diet, but still the people were developing ketosis if they were looking at their blood levels. So I think that first study that we did, we completely overdid it with the ketones because neither Nora or I had really much background in the ketogenic diet. So we just went like, oh, okay, this is the composition. We just need to give the most difficult or the most rigid diet out there that leads to the highest level of ketosis to see a first effect. But I think that study could easily be done with a modified Atkins diet, and we would see the same effects. Because if we look at the millimolar levels of ketones in that first week, where we saw a strong effect on withdrawal, the millimolar were only around like 1, and then we went all the way up to 4. Why? We didn't need it.
Maybe you should test this in rats before you do all that work in humans.
But I think milder is better. Also just to stick to a diet, of course, and for the health effects.
My question just got asked, sort of, but that's okay. The question I had was about the utility in the long term, and it's sort of asking you to editorialize a little bit on, is the utility mostly early recovery or kind of post stabilization from withdrawal? And then maybe, if you don't mind, how does that compare to things like naltrexone in terms of utility, pros and cons?
So, I mean, one of the reasons that people relapse are withdrawal symptoms. I think withdrawal and alcohol consumption longer term are quite related. But it looks like the ketones could be very beneficial. At least we've shown this in humans during this acute withdrawal phase, but we still have not shown that a ketogenic diet or a ketone supplement actually leads to reduced drinking. We just haven't done the studies yet. But ideally, for example, with the ketone supplement, if you just acute effect is you can absorb alcohol, it might be clinically useful for people who are at risk, like, oh, I walk past the bar and I think I'm going to relapse. Just okay, drink a ketone supplement, and immediately you can't really get the effects of alcohol. So that almost parallels more with like an end abuse, where you only get negative effects of a drug. But I think maintaining a ketogenic diet may just lead more to reductions in craving altogether. If that answers your question. And I honestly haven't done the longer term ketogenic diets because it's going to be so difficult to monitor people and to make them stick to a diet. There are now companies where you can just send diets to people's homes. So that's what I would like to do. And then monitor. There are these ketone or continuous glucose monitors, continuous ketone monitors, where you can actually monitor it for 2 weeks in a row. So you can actually do the trial and then afterwards see whether there were glucose spikes and whether ketone levels were high, whether people were cheating, and also if reductions in alcohol consumption, if they correlated with glucose spikes, for example. That would be so cool to do.
Thinking about the preclinical data you showed early with alcohol dehydrogenase levels, and if I remember right, it was animals that were on the ketogenic diet had lower levels of alcohol dehydrogenase. I seem to recall that there are some differences, genetic differences across individuals in humans that, some people express higher levels of alcohol dehydrogenase and it's kind of genetically determined to some extent. I wonder if you've thought about that in terms of kind of individual differences in people. I imagine that it's probably related to alcohol use disorder in the first place, but I also wonder if it's related to kind of the response to this ketogenic diet in terms of, this person has a higher level of alcohol dehydrogenase. Genetically, maybe they are more or less responsive to the ketogenic diet.
Yeah, so people who can't break down alcohol very well could be ADH1 or the second one, that have conversion to acetate, which is more like the flushing symptom, that are protected for alcohol use disorder. So people who can break down alcohol very well are actually more likely to develop an alcohol use disorder. They drink much more to get the same effects. They don't get the negative consequences, or I never get but if you start drinking early, you've drunk so much more over years compared to people who after one glass of wine would already feel it. So being able to break down alcohol fast is a risk factor for alcohol use disorder. So that's also what many NIH reviewers said, well, with this ketone supplement, if it doesn't get absorbed, people may just drink 3 times more and get the same effect in that way. Could giving this ketone supplement actually promote drinking? So yes, based on the alcohol dehydrogenase literature, etc., it could be, but that's not what our animal studies show. And it's also not what our human studies show. Like, no, I didn't like this, but those were healthy volunteers. So again, we need to test it in alcohol dependent population. But I was really fascinated that on both sides of the alcohol pharmacokinetics, like if you get too intoxicated with the ketogenic diet, rats didn't like it. But if you don't get absorbed, rats also didn't like it. So there's an optimal curve. You need to be able to feel the effects of alcohol, to like it, to want it, to have beneficial effects and anxiolytic effects, etc. But if it's too strong, too much. If it's not there at all, why bother? Yeah.
The quick increase of blood level and effects in your brain, that's what drives the addiction. That's so if it makes sense what you're saying, if it's too much, it's too much. And if it's too low, just coming to the brain too slowly, you don't get the kick. So it makes sense, actually.
Yeah, and you see it also with different routes of administration and different drugs, like something like smoking, that's just, it immediately goes into a bloodstream and into your brain, dopaminergic. And then with one cigarette, you have so many exposures of it as well. It's But yeah, I don't really dare to say that that's more addictive than drinking alcohol. But also, like smoking opium is less addictive, I think, than IV, where you just pump it straight into your bloodstream.
That's right. So IV substance use reaches the brain very quickly, and the effect is almost immediate. And if it's a slower absorption, for whatever reason, it doesn't reach the brain as quick. So there's less, it's less likely to cause the addiction and the dopamine response in the brain that we would expect that drives the addiction.
Yeah, and there's some researchers that study IV alcohol exposure, but I wonder how pleasant that is. Maybe for most people, it's not pleasant to get like an immediate kick of alcohol. So also there, that may actually show that same, like, okay, not having it or it's not absorbed, there's no effect. But if it's too quickly, like IV, it may not be pleasurable. Otherwise, we would see people drink or take alcohol IV, right?
Shots. Anybody else?
Thank you so much. These were wonderful questions.
Yeah, thank you, everybody. It was really nice, robust discussion. Thanks for coming. Have a great day.
And I learned so much today. Dr. Shelmy here has diagnosed me with visual snow syndrome. I did not know I had that. And he was describing it as like, huh, doesn't everybody see it that way? So apparently in my world, it's snowing all the time when I look at things, but particularly in Minneapolis. So thank you so much.